All right, this chapter is going to focus on nerve and muscle physiology. So we kind of take ourselves away from kind of like studying movement and forces here to just look at, again, some of the background just physiologically, kind of how our body moves and how, and some of the which the nervous system and the muscles kind of interact with each other to create movement. When we look at movement, so we know that nerves and muscles have to communicate with each other. They communicate at that neuromuscular junction where the nerves come in. And we have, we have two types or two parts to our nervous system. We have efferent and afferent nerves. Efferent are your motor nerves, so those are the nerves that cause, for instance, contraction of the muscles and movement. And afferent are our sensory nerves. Okay, so we have various sensory nerves throughout the body. For instance, our proprioceptors are sensory nerves that give us feedback as to our body position. Okay, and again, they're present in all our soft tissue in the body and in our joints, but both aspects of the efferent afferent nervous system will communicate with each other as we're kind of going through movement. So look a little bit about nerve and muscle cell physiology. So we kind of looked at, in previous classes, we did kind of look at some other, the nerve and how it works. So nerve membranes are both excitable and irritable. So with the fact that a nerve is excitable means that it does have what's called irritability. Irritability is the ability to react to a stimulus. So that's what nerves essentially are. They're excitable because they are irritable. Irritability of a nerve is dealing with the ability to react to a stimulus. So the nerves in our body are stimulated by, for instance, different ion exchange inside the body. So we look to create, when we have an ion imbalance, it creates a potential difference. So when we're talking about nerves becoming active or being at rest, we look at this difference between the inside of the charge of the inside and outside of the nerve. So the nerve has a resting potential. So if you look, and the resting potential has a range, but if you look at the picture on the bottom here, you can see that it's showing a resting membrane potential of negative 70 millivolts. So that's basically the nerve at rest. So what will happen is neurotransmitters. So neurotransmitters are essentially the chemicals that pass information on to the nerves. And what we do is when a nerve, when so much, when that potential changes, that process is called depolarization. So when a nerve membrane, or basically when a nerve is basically responding to a stimulus, it's called depolarization. And that's the point at which the resting potential starts to become more positive. And again, you could see that in the little illustration in the bottom of the screen. As the nerve potential becomes more positive, it depolarizes, and that's usually when you're going to get your response. Then when it gets back to resting potential, you get repolarization. So obviously at some point, the message getting sent to the nerve to fire is going to stop. It's going to get back to resting potential. Now for a little while, you'll also see that you have hyperrepolarization, and that's basically a refractory period. So what that actually does is it kind of takes the nerve to slightly below resting potential, and what that does is in order for that nerve to get stimulated again, the stimulus actually has to be greater. So I'll show that on the bottom part of the screen here. So again, you see your resting membrane potential. So this nerve gets stimulated. So there's an ion exchange that causes that to happen. It becomes more positive and it activates. Then when the nerve goes back to resting potential, it repolarizes. And as it repolarizes, it will actually do this. It will go through this hyperrepolarization. So all this means is that for this nerve to be stimulated again, the stimulation has to be that much greater. So it will go through this refractory period where the only way this nerve can get stimulated again is if there's increased messaging basically being sent to it to change. Okay, so that actually has to be higher than at that point. I mean, eventually it'll get back to its regular resting potential. So again, we look at the nervous system. We have different components to it. We have the somatic and the visceral. So the somatic nervous system is basically responsible for, if we look at our muscles, so like our motor nerves going to our muscles, the somatic, whenever you hear the term somatic, it means structure of the body. So that's kind of like the part of our nervous system that controls like our muscles, our voluntary movement. The visceral nervous system is also known as our autonomic nervous system. The autonomic nervous system controls the parts of our, it controls the functions of our body that we don't think about, that are involuntary, okay? So things like digestion, breathing, our heartbeat, those things. That's all controlled by the autonomic nervous system. So again, the somatic deals with the structure of the body. It's voluntary. It's our ability to kind of do things like movement, voluntary actions, and then the autonomic nervous system is all our involuntary. Obviously, too, we have the central and peripheral nervous system. So the central nervous system is the brain and spinal cord. And the peripheral nervous system is the nerves as they kind of branch out into the periphery. We also have two other components to point out as it relates to the nervous system. We have the upper and lower motor neurons, okay? So you can kind of see here the upper motor neurons. So the upper motor neurons are the portion that spans from the brain to the spinal cord. Okay, so those are the upper motor neurons. So the upper motor neurons are actually located in the cerebral cortex, and they go to the spinal cord. So it's basically, here's your upper motor neurons. And then the lower motor neurons, they're in the ventral horn of the gray matter of the spinal cord. So the lower motor neurons basically go from the spinal cord out into the periphery. So there you're looking at the two different aspects of that, the upper and lower motor neurons. So looking at different types of nerve fibers, we have type A, B, and C, and then we have subtypes of all of those. So you have different types of type A. You have your A alpha. They are both motor and sensory. So, the alpha motor neurons are commonly the ones we think of when we look at skeletal muscle, motor activity. They're efferent as they are motor, but there are also A alphas that are sensory related, and they will activate, again, proprioception, so they do give sensory feedback from that. A beta are strictly sensory. They respond to touch, pressure, and vibration. They're present in the muscle spindles, touch receptors, pressure receptors. A gamma are another motor nerve, so they strictly have motor ability. And then A delta is actually our pain fiber. So A delta is sensory, but they actually transmit pain, particularly pain from acute injury, okay? They also transmit temperature as well, but pain's commonly the thing. Those of you in both athletic training and physical therapy, when you learn about therapeutic modalities and pain and how to stop that, you'll learn about the A delta fibers and the influence of pain from those particular fibers. Before I get into myelination, we'll talk about type B. So type B fibers are autonomic. They're strictly autonomic as it relates to the sympathetic nervous system, okay? And type C are also sensory. They are afferent fibers. And again, type C also transmits pain. Now, type C fibers, we're going to get into myelination, they aren't myelinated. So their conduction velocity, or the speed at which they send information, is a lot slower. So they transmit different types of pain signals than you would get with the A delta. Okay, that takes us into myelination. So all myelin is just a sheath that covers the nerve fiber. So when a nerve has myelination, what that does is it increases the conduction velocity. In other words, the information that gets sent along the nerve is a lot faster. Okay, so that's important in regards to speed of nerve conduction. And you could actually see that if you look at table 3.1 in the book. And you don't have to memorize the conduction velocities. Just know that, myelinated nerves, carry information faster. Obviously, I want you to know that the different the types of A fibers and what a B fiber is and what a C fiber is. But you don't have to know the conduction velocity, but you can see that. And we talked about the upper motor neurons and the lower previously as well. So the upper, they're in the CNS, and they take impulses from the brain to the motor neurons in the spinal cord. and then the lower motor neurons take information from the spinal cord to the muscles. And then that takes us to the muscles. So kind of understanding the structure of muscles. So we look at here kind of a muscle. So when you're looking at this picture, so here's this is the bone. So here's the tendon. So here's the muscle itself here. So you see what we do, and I think most of you have probably seen similar illustrations like this with muscles, kind of looking at the anatomy of them. So here's the muscle in its whole. So again, we look at the different layers of the muscle. First, we have what's called the epimysium. Okay, that's the most superficial soft tissue covering the muscle. It's basically your fascia that covers and kind of envelops the muscle and other soft tissue. So immediately beneath that, you have the perimysium. Okay, so you can see the perimysium. So what we actually do here is we're going to tease out what's called a fascicle. So a fascicle is, if you're looking at this, is a bundle of muscle fibers. So here's a muscle fiber here, okay? So a muscle fiber, when you got a bunch of muscle fibers, they're held together by perimysium inside what's called a fascicle, okay? We break that down a little bit further. The covering of the individual muscle fibers is known as endomysium, okay? So the endomysium covers the individual. So this is a muscle fiber here. Okay, muscle fibers are made up of myofibrils. Okay, so again, we'll tease another part out. This is a myofibril right here. So all these myofibrils are packed inside the muscle fiber that's covered by endomysium. And then inside that, we go a little bit further, are the myofilaments, actin and myosin. Also inside there is what's called the myofibrils. You have the sarcolemma and the sarcoplasm, the fluid inside there as well. So the myofilaments, this is where, if you remember the sliding filament theory, where contraction actually takes place at the muscle. So here we go to the smallest level with the filaments. We back all the way up to kind of the macro level, if you will, with the muscles. And right here is where that active muscle contraction, or what we call the sliding filament theory, takes place. So we can see here, if we kind of break this down a little bit, we can see that the sarcomere is the smallest contractile unit of the muscle. So if we look here, they're teasing out the sarcomere. This is kind of what the sarcomere looks like, okay? So here's your myofilaments, and you can see how they kind of tease them out. They kind of show you what the actin and myosin looks like, okay, coming off of that picture. So the actin is the thin filament, the myosin is the thick filament. And what you look to get in order to create muscle contraction are what are called cross bridges. And cross bridges are what happens when actin and myosin kind of slide over one another and creates that interaction between them that produces shortening of the muscle. So essentially what you have is, again, the earliest thing that happens is calcium gets released. So that kind of triggers the nerves to kind of start the process. And then we end with actin and myosin kind of meeting, sliding over one another, and then that produces the shortening or the muscle contraction. So here is, so if you look at this, so here it is at rest. So you can see in this picture, so if you're looking here, you can see, so here's your myosin, here's your actin. Okay, so the actin's your thin filament, your myosin's your thick filament. And you see the process here, you see the coupling and then the contraction. So what's going to happen is the actin is going to get pulled over the myosin, and that's going to create the shortening of the muscle, okay? You have your four different phases here. Rest, coupling is when they first interact, contraction is the actual shortening, and then recharging is when they kind of go back to their normal resting positions. Okay, muscle fiber types. Again, another thing that I'm sure most of you have encountered before. So you have your type 1 and type 2. So again, when we kind of look at muscle fiber typing and kind of talk about some of the differences between type 1 muscle fibers and type 2 muscle fibers. So type 1 are also known as your slow twitch. Type 2 are known as your fast twitch. So type 1 muscle fibers appear dark when they're looked at underneath a microscope, and part of the reason that is because they contain large amounts of mitochondria and they have a high concentration of myoglobin, which is basically the muscle hemoglobin. That's the oxygen storing component of the muscle. Whereas type 2 tend to appear more pale, okay? They contain fewer mitochondria and only have small amounts of myoglobin. However, when you're looking at the difference between the two, type 1 fibers are good, and if you just think about the description I just gave, type 1 fibers tend to be much more adept to endurance types of activities, whereas type 2 tend to be for more power, fast, quick types of activities. Now, there's two different types of type 2, and this should actually say type 2A, and you have what's called type 2B, or what some textbooks also refer to as type 2X. It gets kind of referred to both in textbooks. So your type 2A are kind of like the intermediate type 2 fibers. So they contain a little bit more of the myoglobin, and they have a little bit more of the mitochondria, as opposed to the type 2B or type 2X, which is more of the pure fast twitch. I've heard some people in some books refer to type 2A as your pink fibers, just because they fall in between the red and the white. Some other things to kind of note about them: the recruitment order. So for normal activities, okay, the size principle dictates that type 1 fibers will get recruited first. So in any activity you do, for the most part, you're always going to recruit type 1 fibers first. Okay, so if you go, say you're even going to lift a heavy weight, say like a 1 rep max or a 3 rep max attempt, your body will recruit your type 1 fibers, but what's going to happen is the type 1 fibers aren't going to be able to respond as needed to conduct that activity, so then your type 2 fibers will come later, and you'll go type 1, type 2A, type 2B, okay? When we look at muscle fiber diameter, type 1 fibers are very small. Type 2A and type 2B are larger, with type 2B being the largest. That's why, again, when you look at certain athletes, depending upon what they do, you can just look at the picture here. Okay, you have a picture of a marathon runner versus a sprinter. Again, a marathon is obviously a long duration endurance activity. Type 1 fibers, they're not going to be as big, whereas you look at most sprinters, both male and females have, a good amount of lean muscle mass, lean tissue on them from, and again, the training and their body type is going to create that as well. Speed of fatigue, obviously, is going to be faster with the type 2, what's going to be fastest with the type 2B. Type 1 obviously does not fatigue very fast at all. Again, that's because they're made for endurance activities. Rate of force development is going to be the best with type 2, particularly type 2B, okay? So again, it's just a matter of, you look at type 1, you look at type 2. Type 1 is your smaller fiber. It's more endurance-based. It has less fatigue to it, and type 2 is more speed, power, force-based. And again, the matching characteristics associated with it. So we look at the motor unit, and the motor unit is the motor nerve, and the fiber or fibers, which it is in most cases, it innervates. So if we look at a motor unit, this picture is demonstrating what a motor unit is, okay? So here's your nerve cell body. These are the dendrites. So the dendrites bring information to the cell body, and then the axon, and this is a myelinated axon. You can see the myelin sheath along here. And then you have your motor end plates, and then here you have a motor unit with essentially 3 myofibrils connected to it, okay? So it's the motor nerve and the fiber or fibers that it innervates, okay? And again, we have motor neurons in the spinal cord. A majority of the nerve classification for skeletal muscles are A alpha. Then we look at and again, there are A alpha motor neurons. The ratio of muscle fibers to nerves is going to be based on the amount of coordination that the area requires. So, for instance, when you have take a muscle, take like your quadriceps, okay, and your thigh. The thigh is used, the quadriceps is used for more big gross motor activities, okay, as opposed to the small muscles in your hand. The small muscles in your hand have to do very precise, very delicate movements. So when you break that down, there's going to be fewer nerve fibers, or I'm sorry, fewer muscle fibers to each nerve fiber in your hand versus in your thigh. Okay, in other words, the motor units in your thigh are going to have more muscle fibers in it because there doesn't need to be precise contraction with that. Whereas when you look at the muscles in your hand, there's going to be more individual motor units in the muscles in your hand because of you having to do much more coordinated and delicate movements with your hand. And motor units in the body activate on the all or none principle, meaning that the motor unit gets completely stimulated or it does not get stimulated at all. So for instance, when you're looking at this motor unit, there's either going to be enough information to get all of these fibers to contract, or it's not going to work, or it doesn't work at all. Okay, and then the greater the activity or the more strength that's needed, you recruit more motor units in order to carry out that activity. So again, still looking at the motor unit. Okay, again, that's what I was kind of ending with this. We talk about the gradation of strength and muscle contraction because we know we don't need the same amount of muscle contraction for every type of activity. You don't need the same type of muscular contraction and force to pick up your backpack as you do to deadlift 300, 400 pounds, right? So what we do is our body knows we recruit only what we need in order to do that. So the first thing is we look at the size principle. We talked about the size principle before. Always your smallest units are activated first. So a lot of those type 1 fibers will be brought into the contraction. So you'll always activate your smallest units first based upon how much force you have to apply. Okay, so if you don't have to apply as much force, you'll recruit those small units initially. Then you look at what's called the recruitment principle. So that looks at increasing the number of motor units. So remember, we said that individual motor units, they either fully contract or don't contract at all. When you need greater force or greater muscle contraction to take place, what you do is you recruit more motor units, okay? So the all or none principle doesn't mean that a muscle has to contract fully or not contract at all. It's just an individual motor unit. So again, if you need more force, you then recruit more motor units. That's the idea behind it, okay? So all those motor units in whatever muscle you're trying to recruit maximally, you'll recruit more motor units. And then you have the excitatory input and the rate coding principle. So what this does is it increases the frequency of stimulation as the time muscle fibers develop maximum tension. So if the muscles are really under maximum tension, what the excitatory input rate coding principle says is your nervous system will actually increase the frequency of stimulation. So again, you're getting as much force as you need to carry out whatever activity it is. So now we look a little bit more at sensory nerve fibers. So our joint receptors, so if you look at this picture here, so this is a picture of the knee, and you can see all these little different fibers, these sensory receptors in our joints and in our ligaments and the soft tissue kind of surrounding the knee. And the reason why we have this is they give us feedback as far as our joint position is concerned. So we have various types of stretch and pressure receptors inside our soft tissue to give us information back in as far as what position our joint is. So for instance, when you're sitting in a chair, you know what position your limbs are in without having to look at them because of the feedback that you're getting from your joints. So when you have your knee flexed to 90 degrees, you know what that does to the soft tissue, it gives you an idea as far as what position your joint is. So it's constantly giving you feedback as far as what position your joint is in. And this is helpful to us in as far as, again, sensory feedback and motor activity, they talk to each other. So, when our joints and the muscle activity around the joints, both of these will communicate, in situations where muscle contraction has to come in to keep a joint from getting injured. And that's why it's so important when a joint is injured, you go through and you do proprioception in rehabilitation, because that will assist with that process. Some very important sensory fibers that are in our muscles and tendons are the Golgi tendon organ and the muscle spindles. So you'll hear about these on more than one occasion because they have a lot of different functions. So again, look at the picture. You can kind of see anatomically where they are. So the Golgi tendon organ is obviously in the tendon. The muscle spindle resides inside the muscle. So these are real easy to get as far as what they do. So the Golgi tendon organ's function is to detect force and tension. It's regulated by autogenic inhibition. Golgi tendon organs respond to tension by causing relaxation. So what Golgi tendon organs actually do is they actually shut the muscle down. The idea being is to help protect it from injury, okay? Again, they're regulated by autogenic inhibition. So what that is, it's inhibitory input to an agonist and excitatory to an antagonist, okay? Again, what they do is cause a muscle to kind of shut down, okay? Because what happens is force and tension builds up, the nervous system senses that, and what they don't want to have happen is for the muscle to tear. So it's kind of a protective mechanism, if you will, okay? Now, again, it's protective beyond what it necessarily needs to be, but it does that nonetheless. Then you have your muscle spindles, which contain what are called intrafusal fibers. So the intrafusal fibers are the specialized nerve fibers, and they're the actual sensory receptors. And there's actually motor function to muscle spindles as well. Muscle spindles respond to an increase in length, but they respond by contracting, okay? So muscle spindles respond to a stretch of the muscle, and they respond by producing contraction, okay? If you look at this picture, so what the picture is trying to show, so it's just the simple activity of the individual bringing a cup to their mouth, okay? So obviously the agonist is the bicep, the antagonist is the tricep. And again, essentially what that's doing is sending that afferent information back upon what we talked about to make sure that the agonist can contract and the antagonist could relax in an activity such as this. Here's another situation where you could see this at work, okay? So this is actually the muscle spindle working here. So here's the muscle spindle. I'll put the highlighter on so you can see. Here's the muscle spindle, okay? So the muscle spindle is a sensory nerve. It goes into the dorsal horn of the spinal cord. It interacts just within the spinal cord, so this information doesn't go back up to the brain. It comes out of the ventral horn, and that's what creates the contraction. So if you look at this, so if this person was just here and someone drops a weighted ball into their hand, what happens? Well, the ball hits, they actually will get some involuntary contraction. What's actually going to happen is when the ball hits their hand, if it happens suddenly, they will actually get contraction. So the contraction point to here is actually involuntary. What it does is that the stretch that gets put on actually causes almost like an involuntary contraction. When you go to the doctor and they do a physical or they do a neurological exam, one of the things they do is they test your reflexes. Well, that actually does activate the muscle spindle, because when the reflex hammer hits your tendon, what it does is it puts the muscle on a slight stretch. The idea is it responds by contracting, okay? So the muscle spindles respond to stretch by causing contraction. This is also one of the reasons why when we static stretch, if we're static stretching to improve range of motion, we do it slow, we don't do it fast. Because if you do it fast, you could potentially end up activating the muscle spindle, where instead what you want to do is activate the Golgi tendon organ. You want to work it slow and have it build up tension, so the Golgi tendon organ actually works in order to inhibit the activity. So kinesthesia and joint and proprioception. So we'll talk about kinesthesia first. So again, kinesthesia gets back to something that we kind of looked at before. Okay, so again, in most situations, a person could be aware of the position their body parts are in, and whether or not a body part is moving still. You're aware of that. That's your kinesthesia, okay? Also known as what we call position sense. Now, position sense usually just refers to more static type of activity. They usually look at kinesthesia more towards like dynamic motion, and position sense more related to static activity. But again, they interact with the whole idea behind proprioception, which is the use of sensory input to determine joint position and movement. Okay, so to kind of break these up, even though they all kind of interact, one more time, just so we're all aware. So kinesthesia is regards to awareness of dynamic joint motion. Position sense just looks at static position. Proprioception is overall just looking at sensory input to determine joint position and movement. Again, this is something very important in physical rehabilitation. Again, for individuals that are athletic trainers and physical therapists, very important as a part of the rehab process, but also strength and conditioning as well. And because again, some of the exercises and things you're going to do are going to help promote this, obviously at a more advanced level, because typically you're dealing with healthier individuals in those cases. Sometimes people might be coming off of injury rehab, and you still will have to know about rehabilitation, but that will be something very important to kind of focus on in rehab. Okay, we look at motor control. So motor control is just dynamic regulation of movement and posture. One of the things we talk about with this is synergy. So when muscles work together to produce movement, we have particular muscles within certain movements that are synergist, and they help to assist with the movement. So again, if you go back and look at your functional anatomy of prime movers and you have synergists. Synergy is important because it allows for movement to be more efficient. Now, where it can become a problem is when synergists become prime movers. We don't want that to happen, and we'll get into why that is a problem later on. But muscle synergy is important, just kind of allows, again, for more efficient movement. So we look at motor control at the spine. We have certain things that in regarding to automatic control of movement as well. We've already talked about reflex motion. So reflex motion is going to take place at the skin, the muscle, and joint receptors. We already talked about, kind of that automatic reaction that will happen with the muscle spindles when they're activated. And then we have, more complicated things like pattern generators, and these are actually modified by cortical commands. So, for instance, pattern generators will dictate things such as walking, okay, so complex activities like that. And that's going to get modified by commands up in the higher brain center as well. So that's an important contributor to our overall motor control. So we do obviously have our voluntary movement, which we do, but there's also this automatic control that will happen at the spine as well. So we look at motor control at another level at the brainstem. So you can see your brainstem here. Okay, so the spinal cord kind of officially ends here. You have the brainstem, the medulla oblongata, the pons, the midbrain. Our automatic control, it primarily deals with our postural control. So our postural muscles during activity respond to efferent information from this area. And here's where we also have contributions from our upper motor neurons. So those motor neurons that go between our brain and our spinal cord are involved here with motor control at the brainstem. And then we have our cerebral motor centers. So these are in the higher areas of the brain. We have our sensory motor system, and this needs to have constant influence from both, because we're constantly getting sensory and motor input to the brain. So our sensory motor system is responsible for that. I'm just going to go over different areas that I just kind of generally want you to be responsible for what they contribute to this. So the pyramidal tract, that's the motor area of the cerebral cortex down to the spinal cord. You have the motor cortex, which is voluntary control of movements and also has cognitive functions. And this is where our motor engrams are stored. So when you're thinking about, kind of like automatic physical activity that your body gets good at that you don't have to think about, those are your motor engrams. if you play a sport, the first time you play a sport and you're kind of learning the skill behind something, it feels a little awkward, but over time, you get better at it and you don't have to think about things as much. Well, those are motor engrams. Those are kind of patterns that are stored, and they're stored in the motor cortex. The cerebellum is just an overall coordinator, so kind of coordinates our motor information. And then you have the basal ganglia, so that's control of both automatic and voluntary movement. It's also responsible for posture and muscle tone, and the initiation and execution of slow movements. Just to maybe connect the basal ganglia to something you may be familiar with if you're familiar with Parkinson's disease. Parkinson's disease affects the basal ganglia. So if you look at individuals that are, unfortunately suffering from Parkinson's, you notice that, their voluntary movement is not as smooth. Their initiation of movement and execution of slow movements is altered. And the reason for that is because that area, that disease negatively affects the basal ganglia. And here are your learning outcomes.